Microbial diversity associated with freshwater aquatic habitats and resident species plays an essential role in ecosystem and organismal health. It is critical to understand the functional roles of microbial communities to better understand microbial-based ecosystem services, in other words, the metabolic roles of microbial communities in larger ecosystems. Our project aims to explore the metabolic functional differences in bacterial communities present on the shells of freshwater turtle species in Oklahoma. From our previous work, we demonstrated consistently high taxonomic diversity in 16S communities associated with shells of red-eared slider turtles collected at sites in Oklahoma and southeast Iowa, although diversity was lower on shells compared to submerged environmental substrates such as woods and rocks. For our current work, we assessed metabolic functions of sampled 16S turtle shell and environmental communities using the bioinformatic platforms QIIME2 and Ivy Kodak.
Using QIIME2, low-quality regions of the sequences were removed. De novo clustering of the data at 99% identity was used to create 99% operational taxonomic units. Taxonomy was assigned using the Silva V13816S database. Using Ivy Kodak, preliminary metabolic functional analysis was performed using the Global Mapper application under default settings. This feature was used to infer the functional profiles of microbial environments using 16S microbiome datasets.
Our ordination analysis supports some overlap in metabolic roles of turtle shell and environmental substrate microbial communities, although shell-associated 16S communities generally featured less complex metabolic networks. Top functions analysis showed that variability in microbial function was greater in turtle versus environmental samples. Using differentiating functions analysis, we recovered significantly differentiated functions for turtle shell communities at a pond site adjacent to a small livestock operation, including for several antibiotic synthesis genes. Finally, using the microbial community metabolic networks analysis, we were able to support that metabolic networks are generally more complex in microbial communities of submerged environmental substrates than for turtle shell communities.
Our general results likely reflect behavioral patterns of turtles, for example, regular basking and drying periods or scut shedding, and are consistent with patterns in taxonomic diversity from our previous work. Local commercial and agricultural activity may also impact microbial metabolic functions, including possible health consequences regarding antibiotic metabolic activities. Our future work will focus on increasing species diversity in shared habitat turtle sampling and exploring 16S or eukaryotic microbial metabolic diversity.